You know, right now, inside your body, there's a silent war going on. It's incredible. You have this amazingly sophisticated army of cells on constant patrol, defending you from all kinds of invaders like viruses and bacteria. So let's dive into that, because today we are going to uncover the secret instruction manual that builds this army, one tiny soldier at a time.
And that really gets to the heart of the mystery, doesn't it? It all kicks off with one single, undecided stem cell. I mean, how does it know what to do? What tells it, hey, you're going to be a hero, a defender of health? And more importantly, what happens when those instructions get all scrambled, turning that hero into a villain like cancer? This is the high-stakes journey we are about to map out.
To really get this, we're going to follow the life story of one of the immune system's most important soldiers. So let's meet the hero of our story, the B cell.
So what exactly do B cells do? Well, think of them as your own personal, highly specialized weapons factories. When some enemy pathogen shows up, it's the B cell's job to design and just pump out millions of these targeted missiles we call antibodies. These little guys are what neutralize the threat and keep you healthy. They are, and I mean this, absolutely critical to your body's defense force.
But here's the thing. A B cell isn't just born ready for battle. Oh no, it has to go through this super rigorous training program. It all starts as a kind of blank slate, a hematopoietic stem cell, or HSPC. Then it makes its first big career choice, becoming a lymphoid progenitor. From there, it fully commits to the B cell life as a pro-B cell. Then it goes through a crucial quality check as a pre-B cell. And finally, it graduates from the bone marrow as an immature B cell, before it gets sent out on patrol as a mature, totally battle-ready soldier. This isn't random at all. It's a highly controlled step-by-step process.
So the big question is, how does the cell know how to follow all these steps? Scientists knew this training program existed, but they didn't really have the full instruction manual. It was time to go find it.
And this was a huge problem. For years, most of what we understood about this whole process came from studying mice. And look, that's helpful, but it's not the same thing. To truly understand human health and human disease, scientists had to get their hands on the specific, detailed, human instruction manual for building a B cell.
To crack this, they used a really brilliant technique called multi-omics, and this slide breaks it down so well. Just think of your DNA as a giant instruction manual, okay? So chromatin accessibility basically tells scientists which chapters of that manual are physically open and available to be read. But, and this is the key part, gene expression tells them which of those open instructions are actually being read out loud to build the cell, like right now. By looking at both, you get the whole picture. You see both the potential and the action. It's genius.
And by using this powerful toolkit, guess what? They found it. They finally uncovered the master blueprint that guides a stem cell through its entire journey to becoming a fully fledged B cell.
This blueprint is what scientists call a gene regulatory network, or GRN. Now, this is not just a simple to-do list. It's a huge, interconnected network of commands. This complex web of if this, then that statements that directs the entire construction process. It makes sure every single step happens in the right order and at the exact right time.
And this illustration is perfect. Within this network, you have these key molecules called transcription factors. The best way to think of them is as project managers. It's their job to find the right instructions, which are the genes, and flip the switch to turn them on or off. This blueprint is basically the organization chart that connects every single project manager to the specific tasks they control.
Okay, I know that sounds a little abstract, so let's zoom in a bit. The researchers decided to put a spotlight on one of these newly identified project managers, and what they found really shows you just how powerful this network is.
The research really highlighted a transcription factor called ELK3. Now, we knew about it before, but its central role in the early stages of human B-cell development, that was a major new discovery. It turned out to be one of the most important managers on the entire construction site.
And just how important is it? Get ready for this. The study found that this one single molecule, ELK3, acts as the project manager for a staggering 2,633 different genes. Think about that. That is the kind of massive influence we're talking about here. Just one molecule controlling a huge portion of the entire cellular machine.
So, what's on ELK3's job description? Well, it's super active during that pro-B cell stage, when the cell is really committing to its career path. Its main jobs are to control the cell cycle, basically telling the cell when to divide and how fast to grow, and to push that cell along the assembly line to the next stage of its training. It's a really critical gatekeeper for the whole process.
Which brings us to the ultimate payoff of all this work. See, this isn't just about understanding how a healthy body works, it's about figuring out what happens when the whole process breaks down and how we can learn to fix it.
So why does having this master blueprint matter so much? Well, because for the first time we have a detailed map of the normal process. And you can't figure out why something is broken if you don't know how it's supposed to work in the first place. That map is the key to understanding what's going haywire in diseases like leukemia.
You know, a really powerful way to think about this cancer, B-cell acute lymphoblastic leukemia, or BAL, is it's a disease of development. The cancer cells aren't some new alien creature. They're just would-be B cells that got stuck. The assembly line just stopped. And these cells are frozen at a specific stage of their training, dividing over and over again, completely out of control.
And this is the groundbreaking part. Using their new blueprint, the scientists could actually map different genetic subtypes of this cancer to the exact stage where the cells get stuck. For example, they saw that leukemia is caused by one specific genetic error, this ETV6 RUNX1 fusion. Those cells get frozen at the pro-B cell stage. But cancers with a different error, like TCF3 PBX1 fusions, they get stuck later on at the pre-B stage. It's a direct link between a specific cancer and a specific failure on the B-cell assembly line.
And this is so much more than just an academic exercise. This map is a revolutionary tool. By knowing exactly where in the development process a patient's cancer got stuck, doctors can start to develop much more precise, targeted treatments designed to attack the specific vulnerability of that frozen cell type. It is a massive step towards personalized medicine.
And that leaves us with a truly fascinating final thought. We now have the detailed blueprint for building a perfectly healthy B cell. We also know where on that blueprint the process breaks down and creates a cancer cell. So the big question for the future is this: could we use that blueprint not just to destroy the broken cell, but to fix it? Could we unfreeze it and tell it how to finish its journey and become the healthy soldier it was always meant to be? The answer to that, well, it could change everything.